So we're going to start off with 6 flash talks from researchers at the University of York. So they're going to cover a wide range of topics of microbiology and showcase just the breadth of research that takes place in York. We're going to do this talk's just 5 minutes long. So we'll give a brief introduction. We're going to do them back to back. But if you have any burning questions or any questions you want to ask the speakers, we really encourage you to type them out in the chat. And if we have time, depending on how we're doing, we may read out some of those questions to the speakers at the end. So make sure you put the speaker's name at the start of your question. But if we don't, we do encourage the speakers to either ask for email addresses or type answers, either in direct messages or in the chat as well, if we have to continue straight on to the next set of talks. So without any further ado, I'd like to introduce our first flash talk, who is Nicholas McGregor. He's a postdoc working with Gideon Davies, talking about xyloglucan-degrading bacteria. So let's start.
Thank you, Mike. Can you hear me?
Yep.
Perfect. And thank you to Christoph for that tribute. We'll miss Will at the YSBL Journal Club and Lab Talks. So yeah, as was mentioned, my name is Nick McGregor, and I've been working with Gideon Davies up on K2 for the last couple of years. I'm a postdoc, and my work centers around new techniques to identify enzymes that break down plant polysaccharides. So I'm going to be talking today about some work that I started back in the fall, working on identifying xyloglucan-degrading bacteria. So this here is xyloglucan. It's a polysaccharide found in the cell wall of all land plants, where it's bound tightly to cellulose. The structure consists of a linear beta-1,4 glucan backbone, which you can see here, with alpha-1,6 xylose branches, which can be further extended with more carbohydrate residues. And these branches prevent self-association, making this a water-soluble polysaccharide. So it's kind of nice to work with. Enzymes that degrade xyloglucan are found in all kingdoms of life, and they've been known for several decades. Bacteria are particularly interesting because they possess a variety of sort of complete xyloglucan-degrading systems. So shown here is a model of a xyloglucan-degrading system from a human gut bacterium. And you can note there's a lot of different glycoside hydrolases here that break down the different glycosidic bonds. And one I want to really note here is this endoxyloglucanase attached to the outside of the cell. So this is the enzyme that's really unique to xyloglucan degradation. So my goal is to detect and identify xyloglucan degrading bacteria using this particular enzyme as sort of a key marker, a molecular marker. And to do this, I wanted to use activity-based probes. So these are covalent inhibitors, such as cyclophelitol shown here, which have been modified to have a detection tag so that when they react with an enzyme, you can then detect which enzyme they've reacted with. So I designed a probe shown here, which I thought would sort of represent the minimum recognition structure of xyloglucanases with resistance to exohydrolases. And this probe was synthesized by the Overcleaved group, some great collaborators of ours in Leiden. And I was able to show crystallographically that this is actually a perfect mimic of the xyloglucanase glycosyl enzyme intermediate. So in yellow here is the true xyloglucanase glycosyl enzyme intermediate, and inside is a complex that I was able to generate using our probe. So we know that this is working like xyloglucan in the active site, which is nice. And I was also able to test the specificity of this using a known model soil saprophyte. So in this case, I grew a bacterium on glucose, cellobiose, or xyloglucan. And within full cell lysates or whole cells, I'm able to see labeling of xyloglucanase exclusively when grown on xyloglucan, and that's the only enzyme that is labeled. So we have a nice probe here that is both reactive and selective. So with that probe in hand, I collected a soil sample from Walmgate stray and cultured a microbial extract on either xyloglucan or celery fiber. And then those bacteria, I tried to isolate them on solid medium and grow those in liquid culture. That, of course, proves to be a bottleneck, as I think a lot of the people here will know. But I was able to isolate, or get at least one isolate which grew reliably on a minimal medium containing xyloglucan and expressed high levels of an apparent xyloglucanase. Genomic DNA sequencing revealed that this isolate was actually a mixture of both a flavobacterium and a sphingobacterium, which could not be grown in isolation. The two of them seem to only be able to live in the minimal medium together. But I was still able to identify the key xyloglucanase using a probe that instead of a fluorophore has a biotin tag on it. So I biotinylated this enzyme, and I was able to pull it down out of solution, out of that lysate, and determine that it is this enzyme here, excuse me, and that it is actually part of a flavobacterial xyloglucan-degrading operon with various new and interesting enzymes. So what I've shown here is that our new xyloglucanase probe is able to, or we can use that to identify xyloglucan-degrading bacteria from environmental samples. And looking forward, I'm interested in applying this workflow to more interesting biosamples and more complex systems, perhaps metagenomics or metaproteomics. So with that, thank you all for your attention, and I look forward to any future questions.
Thank you very much for that talk. So our next speaker today is Rory Elston, who is a BBSRC White Rose PhD student working with Gavin Thomas and Danny Ungar.
My project is titled Understanding and Exploiting Bacterial Sugar Transporters for Increased Production of Prebiotics. The prebiotics in question are human milk oligosaccharides, or HMOs. They are a group of glycans found in human breast milk that are metabolized by specific species of bacteria that are found within the infant gut. The most prevalent species are from genera the bifurcating and lactobacillus. These species are able to metabolize HMOs because they contain genes that encode import proteins and glycosyl hydrolases capable of respectively importing HMOs and breaking them down. The metabolism of HMOs by these bacteria produces short chain fatty acids, which convey anti-inflammatory effects on the infant host. The HMOs themselves have also been shown to convey a wide range of immunological benefits to infants and adults. To infants, sorry. This project has an industrial partner, Glycom, who are world leaders in the production of HMOs. At a commercial level, they're based in Denmark, which is an important research centre for HMOs, and they're involved in understanding how HMOs are beneficial to both infants and adults. The current manufacture of HMOs is through wholesale biotransformation of E. coli. Currently, there's a bottleneck in the production using this technique, which is the efflux of HMOs from the bacterial cytoplasm into the extracellular medium. This is really important because the inhibition caused by this bottleneck, this inefficiency, causes feedback inhibition in the production of HMOs within the E. coli and can cause cell lysis. In addition to this, the general isolation of HMOs from the extracellular medium is much more simple than from isolating from inside the bacterial cytoplasm. So understanding understanding this process will hopefully improve the export and overall efficiency of HMO manufacture. We do not know which sugar transporters are responsible for HMO efflux, again showing why research in this area is really important. Bacterial sugar export in general is really understudied. Only a limited number of sugar export transport proteins have been identified. The best understood are the sugar efflux transporter subfamily of MFS transporters, or SET subfamily, which are present in E. coli. They've got 3 members, SET A, SET B, and SET C, which share a sequence identity of over 45%. SET A is easily the best characterized in terms of its physiological role and its substrate specificity. Its substrate specificity, I've shown below, was determined with a competitive transport assay. It recognizes hexosugars, disaccharides, glucosides, and galactosides, unable to recognize non-hexosugars, phosphorylated hexosugars, and the trisaccharides maltitose and raffinose. Importantly, a company which produces HMOs on a commercial scale, similar to how Glycom does through E. coli, published a patent saying that SET A is capable of free fucosyl lactose export, which is a trisaccharide HMO. The fact that SET A seems capable of HMO export shows that more research into SET A is needed to see if it's capable of exporting different HMOs, and given the similarity between the 3 transporters, this should be extended to SET B and SET C as well. As stated, the physiological role SET A plays is well known as well. It's involved in the bacterial, or the response of E. coli to sugar phosphate stress. Sugar phosphate stress occurs when high levels of phosphate sugars are reached within the cytoplasm of the bacterial cell. This occurs when genes encoding glycolytic enzymes are mutated. Set-A is in an operon with the gene that encodes Set-A is found in an operon with the gene encoding the small RNA SGRS. It's a really interesting RNA molecule. It's able to base pair with the help of the RNA chaperone HFQ to the mRNA of PTSG and ManXYZ, inhibiting their translation. They produce the phosphotransferase systems PTSG and ManXYZ, which are responsible for glucose and mannose import and phosphorylation. So by reducing the levels of these proteins, you reduce phosphosugar concentration. It's also able to be, I said it was interesting, the small RNA is able to be translated. It contains a small open reading frame, which produces the microprotein SGRT, which is able to bind to the PTSG phosphotransferase system and import glucose import and phosphorylation. The role Set-A plays in this is that a phosphatase called YGAL dephosphorylates the sugar phosphates and Set-A effluxes the sugars. The functions of Set-B and Set-C are completely unknown and further research is warranted. The current plans are we want to study cis-set transporters to try and understand if they are able to efflux different HMOs in general, because this would improve the efficiency of HMO production, but we also want to see the metabolites they are able to transport, as this will give clues to their in vivo function. Transporters will be produced in E. coli and purified. We're going to use a thermal shift assay to try and screen a wide range of metabolites and HMO molecules to see which molecules are recognized by the set transporters, and then perform a competitive transport assay in reconstituted proteoliposomes to try and gain more information about their abilities of the different transporters to transport various molecules. Yeah, I just want to say thanks to Gavin and Danny, to my industrial partner supervisor Manos, and to the members of the Thomas and Ungar research groups. Thank you.
Thank you very much. So next up, we've got Chris Furlan, who's doing a PhD with Caroline Descent in the chemistry department.
Sorry for the delay. It's with Jamie Blaza.
Oh, sorry, I was wrong.
Yeah, that was my previous supervisor.
Apologies.
Okay, my PowerPoint seems not responding. Okay. Can everyone see my screen?
We can see it, but it's, yeah, yeah, it's working.
Okay, so I'll talk about the Thermotoga maritima hydrogenase, that it's an electron bifurcating enzyme, and I will explain that later. So it's derived from Thermotoga maritima, that is an anaerobic hyperthermophilic bacteria. It's an iron hydrogenase and uses both NADH and pyridoxine to reduce H+ to hydrogen. So why do we study those hydrogenases? Because they are implied in hydrogen production. So the hope is that in the future, microbial communities can be engineered to retrieve energy from waste material. And hydrogenase play a really important role in this. So I'm showing here why the hydrogenase I'm presenting is so different. Regular hydrogenases, they use just the one redox partner and to reduce hydrogen. So there's an iron sulfur-cluster chain and the electrons are transferred to the H cluster. But with bifurcating hydrogenases, you have two redox partners to reduce hydrogen. And one reaction with the proton is thermodynamically favorable and the other is not. So what happens is that one favorable reaction is pushing the other one. And that's the peculiarity. So there's a lot of confusion about its mechanism and many proposals. So we thought that with cryo-EM we could exclude some of the mechanism and understand the system better. For people who are not familiar with the technique, the protein sample is quickly frozen in ethane, and you produce a thin layer of vitreous ice, and then electrons are used to take many micrographs that are averaged together. At the end, the structure obtained looks like that. So it's a very complicated complex with lots of subunits and with a very complicated electron transport chain. In addition, when the map threshold is changed, you can see there's a sort of blurry density, and we call that density bridges because it looks like a bridge. So to get a better map with that structure, we used a approach, a data processing approach called the symmetry expansion. And after using that approach, it's clear that there's a bridging density between the two lobes. This bridging density has also iron sulfur cluster, and it's therefore likely to be implicated in the mechanism as producing and maturating the protein with iron sulfur cluster is expensive for the cell. So also in addition, the B chain of the bridge is moving, so has a conformational change. We don't know yet the importance of it, what's its function, but I think it's likely to be something implicated in the mechanism. So to give a brief overview, that's how complicated the electron transport chain looks. Each of these squares and rhombus are iron sulfur clusters. FMN is flavin, and then there's the H cluster. So it's extremely complicated and definitely not a good starting enzyme for our first project, but also fascinating how complicated this thing is. So to conclude, even if we collected all those data and we got a structure, we are still far from understanding the mechanism. We know the composition of the iron sulfur, the electron transfer chain, but we still don't know where the bifurcation center is. So the center that allows the splitting of the electrons. And we don't know where the ferredoxin bind and so many unanswered questions, but at least some mechanism can be excluded. And yeah, that's it.
Thank you very much for that talk. Our next speaker is Alex Scott. Alex is a BBSRC White Rose PhD student in Christoph Bauman's group.
Okay, thank you everyone for letting me talk. So I'm going to be talking about a chapter of my PhD research, which is to do with quantifying the pore formation in the gram-negative cell envelope using low temperature plasma. So we use low temperature plasma as an attempt to use it as instead of antibiotics. So it's very good at sterilizing wounds and equipment. And the way that it does that is essentially by we produce a strong electric field across a gas jet, and then that ionizes the gas, and then it produces reactive oxygen and nitrogen species. And these are inherently microbial and antimicrobial and are very potent to bacteria. However, we don't really know how that damage is happening. So what I'm looking at is the biophysical mechanisms of this damage on the cell envelope of specifically E. coli at the moment. So some preliminary experiments that we did, we very clearly saw that pores were being formed in the cell envelope due to using the backlight live dead assay, which requires a membrane impermeable dye to get into the cells. So if they're getting into the cells, somehow these pores are being formed in the cell envelope. So we wanted to ask ourselves, how big are these pores? How frequent are they? And actually, does the LPS, the lipopolysaccharide complexity, affect this damage in any way, shape, or form? So the way that we're doing this is using these nanoparticles called quantum dots. So quantum dots are, as they might seem, in the nano scale regime, and they're very small. And the fascinating thing about them is that the semiconductor crystal they're comprised of in the middle, the smaller you make that, the shorter the wavelength emission once excited it is. So you can actually have a variety of these different sizes of quantum dots. So we're using green, yellow, and red at 525, 585, and 655 nanometers respectively. And then we're hopefully trying to see whether if more of the red are fluorescing or more of the green, we can then kind of roughly guess how big these pores that the low temperature plasma and the produced reactive oxygen species are producing. And then we're also varying the LPS length by using 3 different strains. We have the smooth strain, which has the full LPS of the O-antigen. We have the rough strain, which is the wild type, so it has the core oligosaccharide and the lipid A, but it does not have the O-antigen. And then we have the deep rough, which is only the lipid A and 2 KDO subunits. So it seems to make sense that there's less LPS and less kind of depth to the cell envelope, that possibly more damage is going to be occurring because there's just less stuff there. So we went through with this, treated the cells with low temperature plasma, immobilized them inside gel, and then I measured the mean cell fluorescence of God knows how many cells, and in the 3 different LPS lengths, and we get a very positive correlation, which is actually yes, the shorter LPS strains do seem to uptake all of the different color quantum dots more, and there is a small decrease between the small green quantum dots and the big red quantum dots. One of the problems that we had is actually the smaller quantum dots also have smaller emission peaks, so they're more difficult to detect above the autofluorescence cells naturally emit when you stress them. And then in these graphs, you can see that we have QD, which is the quantum dot sample, the LTP, which is cells that are just treated with plasma, no quantum dots added, and then C, which are the natural autofluorescence of cells. And you can see these long tails on all of these mixed scatter box plots, and these are the cells that we have thresholded and calculated, so we've been taking up these quantum dots. So it's really nice to see that varying these LPS does have a difference. Whether more damage is being done or whether the longer LPS chains physically stop the quantum dots from diffusing into the cells, it's a completely different question. And we hope to answer that by not only taking direct measurements of the quantum dots using atomic force microscopy, but also I'm currently producing a complementary leakage assay where we're actually going to be having fluorescent proteins from inside the cell diffuse out. So we get the other side of that coin. So thank you again for letting me speak. Obviously, I want to thank my supervisors and everyone in the lab who has helped me. Thank you very much.
Thanks, Alex. So next up, we have Sarah Tindall, who's a BBSRC White Rose PhD student with Garen Thomas, Jen Potts and Marianne Vanderwold, who's going to be talking about her structural analysis of acyltransferases.
Thank you. Yeah, I'm a final year PhD student in the Thomas lab at the moment, and I'm going to be talking about my work on acyltransferase proteins, which are involved in the modification of lipopolysaccharides. So acyltransferase family 3 proteins are involved in bacteria in a wide range of different functions, including root nodulation, which is important for symbiosis, acetylation of peptidoglycan, which gives resistance to lysozyme, modification of macrolide antibiotics, which increases their efficacy, and acetylation of the O-antigen of LPS, which is important for their virulence. So as you can see from this diagram, acyltransferase family 3 proteins have 10 transmembrane helices, and then some, but not all of them, have an additional estrogen H domain, which is attached via a periplasmic linking region and an 11th transmembrane helix. And as shown here, both domains, where present, contain residues which are essential for activity, suggesting that when it's present, then both domains are required for function. So this is our kind of hypothesis about how these proteins work. So the acetyl group, presumably from acetyl-CoA, although we're not sure about that completely yet, is transported across the membrane by the acyltransferase domain, where it's then transferred onto the estrogen H domain, which can attach it to the acceptor substrate. Although, as you can see from this model, there are plenty of questions that we still need to answer, which is where my project comes in. So we started by looking at the structure, or determining the structure of the estrogen H domain and periplasmic linking region from OFB, which is an acyltransferase protein that acetylates the rhamnose residue of the LPS in Salmonella paratyphi A. And the first things that we found was that it looks quite different to what you would call a normal estrogen H domain. So to start with, it has this additional helix, which gives it an elongated shape that's not seen in other SG&H domains. And secondly, the periplasmic linking region, which was previously assumed to be flexible, actually forms a structured extension of the SG&H domain. And when removed, the extension, the SG&H extension actually decreases the melting temperature by around 10 degrees, which suggests that it's important for stability. So taken together, we know that when you remove the SG&H extension, it reduces the stability of the SG&H domain. And as there are very few residues between the 11th transmembrane helix of the acyltransferase domain and the start of this structured extension in the SG&H domain, it suggests that the two domains are likely to be in close proximity. So to test this hypothesis, we used in silico coevolution, and this uses huge multiple sequence alignments and looks for pairs of residues where when one residue mutates, the other one mutates at a similar time. And these are known as correlated mutations. And these pairs of correlated mutations suggest that they're likely to interact or make a contact in the 3D structure. So it can be used to make predictions about the overall 3D structure of a protein. So we started by looking for interactions between the SG&H domain and the acyltransferase domain. And we found that there were a cluster of residues on the additional helix, which are predicted to form interactions with one of the periplasmic loops in the acyltransferase domain, suggesting that there are likely to be interactions between the two domains. And then we took it a little bit further and used the coevolution to predict interactions between transmembrane helices. And we found a large number of interactions between transmembrane helices 1, 9, and 10, 2, 6, and 5, and helices 7 and 8. And this pattern of interacting helices, we've not found any examples of that in other 10 or 11 transmembrane helix proteins, suggesting that the acyltransferase domain is likely to have a novel fold. So although we haven't dealt with all the questions that we had at the beginning, we've definitely been able to update our working model and get some ideas about the potential overall structure and mechanism of these AT3 acyltransferase family 3 proteins. So just want to thank in particular my supervisors, Gavin, Marianne, and Jen, and thank you for listening.
Thank you to Sarah for that talk, and thank you to all of our Flash Talk speakers for some really interesting and really diverse talks about microbiology. I think in the interests of time, we're going to move on to the next speaker, but if you do have any questions for our Flash Talk speakers, then I would encourage you just to pop them in the chat. If I can ask the Flash Talk speakers to just have a little look there, and maybe if they want to respond to any questions in the chat, then to go ahead and do that.
Okay, brilliant. Yep, thanks again to all our Flash Talk speakers. Absolutely brilliant microbiology. I'm really excited to introduce our next speaker, Chayan Kumar Saha from Umeå University. I'm sure I got none of that right. But Chayan's a bioinformatician in Jenna Atkinson's lab, and today he's going to tell us a little about this really nice tool that he's developed that looks at gene localization and neighborhood conservation, and it's really useful and highly recommended. So please, over to you, Chayan.
Thank you for your nice introduction, Michelle. Can you see my slide?
Absolutely, yeah.
Yes. So good afternoon, everyone. Welcome to my presentation. First, I'd like to thank Christoph for your tribute. Thanks to all the organizers for this event, and thanks for inviting me. Already you know me. My name is Chayan Kumar Saha. I'm a PhD candidate from group of Gemma Atkinson, Department of Molecular Biology, Umeå University, Sweden. An announcement shortly, lab is moving to Lund University later this year. Anyways, so today I would like to talk about my developed bioinformatic tool, webFlaGs, and its web version, webFlaGs, and how webFlaGs can analyze conservation of gene neighborhood. It is well known that genes in bacterial genomes are often organized into operons. Also, functionally related and co-regulated prokaryotic genes can be organized as clusters. A gene cluster can contain multiple operons. Gene clusters encode useful functions such as antibiotic biosynthesis, antiphage defense systems, secretion systems, toxin antitoxin systems, etc. So why is it interesting to study gene clusters? If you have a gene of unknown function, prediction can be made about its function based on where it is encoded. One can compare gene cluster in different organisms to know the evolutionary history. Also, understanding the architecture of cluster can help to design experiments to investigate its regulation. We are mainly interested in the function prediction and the evolutionary history. In the beginning of my PhD, I analyzed more than 24,000 of predicted proteomes across the tree of life and identified protein sequences belonging to certain superfamilies. I will come back to what about these proteins are later. I classified these protein sequences into families and subfamilies and made a database. I will also explain this later. We usually know the function for some subfamilies in our database, but not for all. So as a bioinformatician, I want to use information regarding conservation of gene neighborhood to predict new function. The tryptophan biosynthesis operand shown here is an extreme example. As you can see, it is conserved from bacteria to archaea. Therefore, I developed FLAGs, stands for flanking genes, a Python tool for analyzing conservation of gene neighborhood. One can download FLAGs from the GitHub link provided on the top. So let's see how FLAGs works. FLAGs takes a list of protein accessions as input. Then it looks for genomes from NCBI to get the flanking genes of upstream and downstream. Next, flanking genes are subjected to very sensitive and iterative sequence similarity searching using jackhammer, and thus the flanking genes are clustered into homologous protein clusters. Finally, FLAGs outputs a figure where query genes are in black and the flanking genes are colored and numbered by the homologous cluster they belong to. The web version of FLAGs or webFlags offers option where user can submit a single protein accession or a protein sequence as input. This is the web interface of FLAGs. Right on top, the web address through which one can use webFlags from any corner of the world. For webFlags, there is no necessity of installation or any prior programming knowledge. It has a very user-friendly interface. This tool was published last year in Bioinformatics Journal. Since then, many researchers have been using it. These are the output examples. As I said before, color and number corresponds to a homologous group, and annotation in also it produces annotation in separate text legend file, which somewhat look like this, and it can be viewed as a word cloud like this. So summer project student Maxence, he wrote this script for this word cloud. Also, one can map the result into tree output. So FLAGs can handle tens of thousands of genes and the user decides its dimension. So if user want to look at thousands of genomes, we'll get an output something like that. But this is also possible to look at a larger window of the genome as well. For example, one conserved secretion system shown here. Okay. Now I'm going to tell you how we have used FLAGs in our lab. One of the things that our group is interested in is the PPGPP-mediated bacterial stress response. When the conditions are not favorable for growth, such as amino acid starvation, bacteria can sense the stress and respond by producing PPGPP in the cell, which onsets stringent response. As you can see in the figure, in case of both gram-positive and gram-negative bacteria, this smaller amount PPGPP regulates multiple important cellular processes like transcription, translation, metabolism, etc. It reduces growth rate and redirects resources to deal with and recover from stress. It plays a key role in virulence during infections and tolerance to antibiotics. Cellular level of PPGPP is regulated by a family of protein called Rayleigh's polyhomologs or RSH enzymes. RSHs can have one of these three domain structure. From the top, as you can see, long RSH, which contain both PPGPP hydrolase domain and PPGPP synthetase domain in the N-terminal region. Long RSH is universal across most bacteria. However, many bacteria can have single domain RSH homologs like small ammon hydrolases, which only contain this hydrolase domain, and small ammon synthetases, which only have this synthetase domain. These single domain RSH homologs have a patchy distribution, probably passed around by horizontal gene transfer. It is unknown why bacteria often carry these enzymes multiple times in the same genome and what is their in vivo function. My PhD research aim was to study functional evolution of RSH enzymes, particularly of SASes. Using phylogenetic analysis, I have identified 13 subfamilies of long RSHs, 11 subfamilies of small ammon hydrolases, and 30 subfamilies of small ammon synthetases. 3 in the left was made from alignment of protein-containing PBGBP hydrolase domain, and in the right was made from alignment of protein-containing synthetase domain. In the beginning, I mentioned about some databases. Yes, I have identified 35,616 RSH proteins and classified them into subfamilies and store information in databases. So 30 subfamilies of small ammon synthetases, and we just know how RelQ is regulated, but we don't know its biological role in the cell. And also, what about others? From FLAGs analysis of identified RSH subfamilies, surprisingly, we observed that SASes can be found in conserved, almost always overlapping 2-gene architectures. In this figure, SASes are in black pentagon, and there is clearly something else in the downstream which is very well conserved. It reminded us about toxin-antitoxin system, or TA system. So TA systems are small genetic modules that encode a toxic protein whose overexpression leads to growth arrest, and an antitoxin that neutralizes the activity of toxin. TAs are typically encoded in bicistronic operon and can be often overlapping, which is exactly what we have observed for SASes and the downstream genes. TAs can have very diverse targets in the cell, usually targeting co-cellular processes like replication and translation. And the antitoxin can have very different mechanism of neutralization. Sometimes it can be protein-protein, sometimes protein-RNA. In general, toxin causes fast reduction in growth rate and readily counteracted by the antitoxin. Most of their functions are pretty much obscure, but some of the verified functions include plasmid maintenance, protection against phages. So my developed tool, FLAGs, predicted that these 5 subfamilies, 5 SAS subfamilies, are encoded in conserved TA-like operons. So here is the experimental validation. This is toxicity neutralization assays done by Steffi from the lab of Vasily Hauryliuk. As you can see in the figure, when toxic pH rel2 is expressed, cells stopped growing compared to the empty vector here. Whereas if the antitoxin is expressed, the cell continues growing. But co-expression of toxin and antitoxin restored the growth. We can see it on the plates, but also in the growth curves. We have observed similar results for additional SASes, although some can be more toxic than others in our system. We can summarize the result in a tree. So taxa, colored in red, is what we have tested and turned out to be genuine TA. Everything in purple is what we have tested and it isn't TA. So SASes aren't toxic when they're found in long conserved operon like RelQ or unconserved operon like RelP. When SASes are in very well conserved small operons, they show toxicity, and thus we call them toxases. Mostly they are 2-gene operon, but one exception here is Toxas Furlan. So as you can see, this is 3-gene operon, and the upstream, the downstream antitoxin here is counteracting the toxicity of Furlan specifically, but the upstream one is a single domain PPGBP hydrolase, which is SH that I mentioned earlier in my slide. It hydrolyzes PPGBP. This antitoxin can counteract the toxicity of all the toxases. Other than that, all other antitoxins specific for their cognate toxins. So HPLC results done by Sophia showed that how expression of Furlan affects the nucleotide pool. It results in dramatic decrease of ATP and GTP and accumulation of PPGPP and surprisingly accumulation of PPAPP. So PPAPP is structurally similar to PPGPP. It blocks ATP synthesis by binding and inhibiting a key enzyme that is important for ATP production. We think that decrease in ATP and GTP pool is contributing to the toxicity. So the story of toxases was published in the Journal of PNAS last year. There is a summary of it. So we discovered 5 SASE subfamilies that are encoded in conserved TLI cooperants, and each antitoxin neutralizes only its cognate toxin, except the one I mentioned in the slide before, the PPGPP hydrolase one, that universally neutralizes all other toxases. And also, the toxicity of toxas Furlan is mediated by accumulation of alarmones, PPGPP and PPAPP, that results in depletion of ATP and GTP pool. In lab, it has been discovered that some toxases don't produce alarmones at all, and they exert their toxicity in a totally different way. Still, our lab is working on that, so we have to wait for some time to hear more about it. So that's the story about toxases, but what about the antitoxins? So what conserved flanking genes do the antitoxins have? So mostly we found that the cognate toxas gene, except for one hypothetical protein carrying a DUF domain. So DUF stands for domain of unknown function. I have shown this figure before. We have to focus on this toxas family PHREL2, specifically this cognate antitoxin carrying the DUF domain that we got interested in. So using the FLAGs analysis, we observed that this domain is almost always in TLI cooperant, paired with different toxin domains. Some of them are known, type 2 toxins like MAZAF or MQSR, and also some are hypothetical. So 12 of them were experimentally validated toxic domains. Taswaki from Gemma's lab have done the experiment. Given the versatility of this DUF domain, we have renamed this domain to Panacea or PanA. Panacea is found in a wide variety of TLOci across the bacteria, archaea, and bacteriophages. Branches are colored by major taxonomic groups in this tree. The rectangles in the inner ring indicate predicted associated toxin groups. Colored circles in the next ring indicates the TA pairs that have been tested in toxicity neutralization assays and the results in those assays. Rectangles in the outer ring shows that the presence and absence of N-terminal domains in PANA sequences. This circular tree here showing just a representation of set of Panacea domain containing protein. So running flags with a number of type 2 TAs suggest there are multiple antitoxin domains like Panacea that can be associated with many toxin domains. We call these hyperpromiscuous antitoxin domains. So the question arises, is Panacea unique in its hyperpromiscuity? So we can find it out with an iterative version of flags, which is NetFlags. Don't worry, you don't have to pay any monthly subscription fee for this. So for now, so NetFlags stands for Network of Flags, Toxin and Antitoxins. NetFlags can automatically identify TLI gene architectures without human involvement. In principle, we can hop with NetFlags from toxin to antitoxin and antitoxin to toxin and continue hopping until we have the full network. The principle is quite simple. Let's say in round 1, we start with an antitoxin A1 and being used as a query. Through flags, we can find a new homolog T1. In round 2, T1 is used as a query. We can find in a similar process, we can find like new homologs A2, which can be used as a query for round 3. So in round 3, as you can see, we found 2 new homologs, T2 and T3, can be used as a query for round 4. So in round 4, we can see that for T2, we don't have any new homolog. We found just A2. So this is a dead end for the program as it considers, because it doesn't find any new homologs. And also for T3, it has been it has been a long conserved operon as incorporated into secretion system, which is very interesting. But as far our program it doesn't consider this longer operon as a TA-like operon. They only conserve, they just only take only 2 gene operons as a TA-like operon. So this is also a dead end for the program. So we can make a network from there, something like this, which is obviously bigger than this in real data set. So this network shown here is the initial step, just starting from pen A, showed just 3 rounds of it. And blue stars here are TA systems verified by our lab, and the yellow stars there, these TAs have been verified by peoples over the years. It's in the literature and toxin antitoxin database, which is kind of proof that the strategy works because we rediscover some previously known TAs. But again, to mention, this result is preliminary and also a bit old too, because last year I was kind of busy with webFlaGs, and yeah, it was really a roller coaster journey with getting banned from NCBI. Well, many memories, but anyways, the real network will be much more bigger than this one, and I'm working on this right now. So in conclusion, I say that flags, we have made flags a tool for anyone who wants to learn more about the evolution of neighborhoods of the genes of interest. And of course, we can help with that. And toxoses are the tip of the iceberg of discoveries we can make about TA across all cellular and viral life. Using toxicity neutralization microbiological assays in the group, we have confirmed 10 predicted novel PAN-A and associated toxin PAN-T TA pair. And for NetFlags, it's still in development, and we are working on verifying new lineages. So with that, thanks to my supervisor, Gemma, for her guidance and support. Thanks to Wesley and all the experimental biologists from Gemma's and Wesley's lab for experimental validations. And thanks for funding. Thanks to funding, and also thank you for listening. If you have any question in your mind, I would be happy to answer them now, or you can write me an email as well.
Thank you, Chayan. That was absolutely brilliant, and that's a great story. And I think this is one of the tools that every molecular biologist should have in their toolbox. So I'm wondering if you could just pop the link to webFlaGs in the chat, if everyone can access it. We did have a couple of questions, but I'll give you one easy one just in the interest of time. Will you be making a web version or open release of the NetFlags that we could use with any starting gene?
Well, that's not the plan. And NetFlags is like kind of, when we run it, we actually get the network out of it. Like we'll get the full network. But also like there is a scope, like we're limiting it in a way, like one gene, one genome or one representative genome from like one strain per species is a way. But you can actually look for like all the species. So there is chance of improvement in there as well, but not actually looking for making a web version for that. Yeah.
Okay, we can maybe have some more questions at the end of the session. I'm just conscious of time, and I would like to thank you again, Chayan. That was really cool. Okay, our next speaker is Dr. Rebecca Hall from the University of Birmingham. I'm really happy to have Becky speak at this symposium. She's a former Yorkie. She completed her PhD here in Gavin Thomas's lab, and have had the pleasure of working on many projects. But one thing I know about Becky, she's a fantastic biologist and interested in evolutionary microbiology. So I'm super excited to hear about her talk that's looking at gene relationships in prokaryotic genomes. So please, over to you, Becky. And
Great, thank you so much. Thank you, Michelle. And thank you for the opportunity to speak to you all today. And can I just say how sorry I am to hear about Will, and my thoughts are obviously with his family and with the L1 community. What must be a really awful time. And thank you, Christoph, for a really nice tribute as well. So I'm going to talk today about the work I did last year in my first postdoc with James McInerney at the University of Nottingham on gene relationships in prokaryote pangenomes. So I want to start by just introducing what a pangenome is. So every time that we sequence a new genome for a given species, we see an increase in gene content variation. So for example, as an illustration, we have a first genome, and then when we sequence a second, we find genes in blue here that are unique to genome 1 and the ones in yellow that are unique to genome 2. And then again, the pink that are unique to genome 3 as we go through. But we also have this set in grey that are core to all of these genomes. And it's these genes all pulled together that form the pangenome of a species. So the pangenome consists of this core genome common to all and the accessory genome that's found only within a subset. And it's the accessory genome that's really interesting, often has lots of interesting adaptations to very specific microenvironments as well. The exact nature of a pangenome varies by species. Some have closed pangenomes where the majority of the genes are core. And what this means in practice is that every time you sequence a new genome, you don't really see an increase in total pangenome size. So for example, for Bacillus here in the plot on the right in blue, with every new genome, the pangenome size stays roughly the same. Whereas other species have very open pangenomes. Streptococcus in orange, every time you sequence a new genome, we see the pangenome size continually increasing because of this large proportion of accessory genes. And it's the accessory genome that the McAneny lab is particularly interested in at the moment, and specifically the relationship between genes in the accessory genome. Does the presence or absence of one gene affect the presence or absence of another within a given genome? And does the order in which these genes arise, does that have an impact on the genes that are found there as well? And to start answering these quite fundamental questions, we look at what's called coincident gene-gene relationships. And so that's the presence or absence significantly of one gene given the presence or absence of another. So just as an illustration, here we have 5 model isolates, and we can say that gene A and gene B significantly co-occur with one another, more often than you might expect by chance. Whereas gene C and gene D, we can say significantly avoid one another. They're not found within the same genome, more often than you might expect. Now, fortunately, we don't have to mine the accessory genome by hand to find all of these. Last year, Fiona Whelan, who's an Anne McLaren Fellow at the University of Nottingham, published CoinFinder, which is a piece of software that does this analysis for us. It takes a gene presence absence matrix that you can make from either Rory or Panaru, and then a phylogeny, and produces an output that includes long lists of these pairs of significant co-occurrences and avoidances. It also details things like the strength of the interaction and gives you a d-value, so you can start to work out whether or not you think this relationship is lineage independent. And what we end up with is networks such as this, where each node within the network is a gene, and they're connected by an edge that signifies a relationship that is significant, either a co-occurrence or an avoidance. And if this is something that you're interested in, we do actually recommend that you use Panaru over Rory for this. And so some of the initial analysis that I did using Rory, we found that it tended to create quite a lot of false positives, particularly for the avoidance networks. Not so much for the co-occurrence, but just something in case you are interested in using it. So throughout my postdoc, it was only quite a short one. We were interested in kind of 3 main questions. First of all, fundamentally, which genes co-occur with one another and which genes avoid one another? And then from that, what does that tell us about pangenome structure and evolution? And then moving on, what can we do with this data beyond simply understanding pangenomes for pangenome's sake? To start answering the first question, we decided to use E. coli as a model system, primarily because we were really interested in really drilling down into the relationships between these genes, what these genes are doing, what role that they're playing. And for that, it was quite important to use a system where we knew as much as possible about the genes that we were looking at. We also had access to a large set of E. coli genomes in Alan McNally's lab at the University of Birmingham. So from this large set, we subsetted out 400 genomes, 20 each from 20 different sequence types. And you can see the sequence types in the phylogeny on the right-hand side here. So we have a selection of multi-drug resistant lineages. We have ST131, ST648, and then a series of commensals as well. So it's a really nice spread across the lineages. When we constructed the pangenome using Panaru, we found that it was very representative of both a typical open pangenome and a typical E. coli. So a small number of core genes and then a large number of accessory genes. And it's this accessory gene, the accessory genome, that we then look at to look for these coincident gene-gene relationships, these co-occurrences and avoidances. And these are some of the networks that we can construct for this. So on the left, we have our co-occurrence network, and on the right, we have our avoidance network. I'm just going to talk to you roughly about some patterns that we see in these networks to start off with. First of all, the co-occurrence network is much larger than the avoidance. We have about 8,000 nodes in the co-occurrence compared to about 3,000 in the avoidance, suggesting that the genes are much more likely to be co-occurring with one another than they are to be avoiding. Both of the networks are characterized by this large connected component in the very center, which is indicative of quite weak interactions. So you might have a single gene that co-occurs with a set of others that don't interact with one another, or you might have gene A interacting with gene B, gene C, and gene D, but not necessarily forming a complete component. For our co-occurrence network, we have around the outside, you can see I've labeled some of them, a lot of perfectly co-occurring components. So within this, we have every gene co-occurring perfectly with every other gene that's present in that component. Whereas for our avoidance network, it's really characterized by what we're calling root excluders. So if you look on the right, I've labeled it number 5, colored in orange. This is a really nice example of this. We have a single gene that's avoiding a whole set of other genes. And then when we dig down into the genes that it's avoiding, we can say that actually these do all co-occur with one another as well. So it's just this single root excluder and that looks quite different and is avoiding all of these other genes. And we can then take these connected components, for example, for our co-occurrence network, and look at the genes that are forming these components, look at what they're doing, ask, are they similar? Do they share function? And that's what we see. So I've started to assign here for you rough functions to these components based on the genes that we find within them. I've highlighted several in pink around here that are related to quite fundamental DNA cellular processes. We have replication, repair, and regulation. And a lot of these as well are enriched in plasmid-related genes, the ones related to maintenance and replication. We have others that are highlighted in blue, that are related to genes involved in secretion systems, different secretion systems based on the component that they're found in. And then interestingly for E. coli, we have several that are related to resistance. So we have one in green that's related to metal detoxification for copper and silver. And then possibly my favorite connected component is highlighted in orange, and this is related to antibiotic resistance. And this component contains several genes related to tetracycline resistance, plus several transposons, and then a metabolite transporter as well. So this is really quite an intriguing component that I'm very interested in looking at. And now that we've started to look at the genes that are within these components, we can then go back to our phylogeny and ask whereabouts are they sitting? Are some sequence types and some lineages enriched for some of these components over others? And so this plot here for you on the right aims to answer this question. We have our sequence type along the bottom and our co-occurrence number down the side in ascending order. And as a reminder, this is out of a maximum of 20 genomes for each sequence type. And I hope that you can see there's a real variation by both sequence type and component in how many times this particular component occurs within a given lineage. Possibly the most obvious one from looking at this plot is component 150, which is one of our ones that's enriched in secretion system genes. And this is found at a really high abundance across the majority of our lineages, with the exception of ST11 and ST3, where it's not found at all. We have a similar pattern at the very top in component number 2, which is another secretion system component. And then our interesting antimicrobial resistance component, again, that's found across the phylogeny, but also particularly in our MDR strain ST648, which is very interesting. And then we have others that are rarely found at all. So 223, a small one at the bottom, which is only found in ST10. And so we've assigned functions to these components roughly based on the majority of the genes that they're found within, genes that are enriched in these components. And so then we started thinking, well, is this one of the main drivers behind these co-occurrence relationships? Are they simply co-occurring together because they share function and share similar pathways? And is this a pattern that holds outside of E. coli as well, or is it species specific? And so to start to answer this, I'd like to segue now slightly away from E. coli for a second into another pangenome, this time of Pseudomonas. And this was constructed by Fiona Whelan. And the network map on the left shows the co-occurrence and avoidance patterns in this Pseudomonas genus level pangenome. These genomes were taken from pseudomonas.com, and the plot in the middle shows the distribution of the genomes within this pangenome. So as you might expect, predominantly they are aeruginosa, but we have some Putidae and fluorescens in there as well. And it's a very, very open pangenome. So the majority of the genes are accessory genes, only a small number of core, which I think is possibly a product of the fact that it is this genus level pangenome. But it does give us a large number of accessory genes to look at. And one of the reasons that we were interested in using this particular database is because the genes in these genomes have Go annotations already assigned to them. And so one of the questions that we asked with this dataset was, if you have a significant gene pair, either a co-occurrence or an avoidance, are you more likely to share Go annotations as a proxy for function than randomly assigned pairs in the accessory genome? And that's what we found. We found that about 70% of co-occurring and avoiding gene pairs do share these Go annotations, compared to about 50% for what we call our abundant accessory genes, genes that we've randomly assigned as pairs in the accessory genome, suggesting that function is one of the main reasons why we have these co-occurrence relationships in pangenomes. And that's all I'm going to speak about this Pseudomonas pangenome for now. But if you are interested, this was on bioRxiv at the end of last year, so please do go take a look, and we'd be happy to take feedback. So now I've started looking at our connected components, we can then move away from that, move sort of down to a deeper level and look at the genes that we think could be most influential in this E. coli accessory genome. And for this, we're looking at what we call hub genes. And these are our most highly connected genes within each of the networks in terms of the number of gene-gene relationships that they form. And so you are a hub gene if the number of gene-gene relationships that you form is 1.5 times the upper IQR number of gene relationships. And we can see here, I hope, in this plot that we have many more gene hub genes than we do in the co-occurrence network than we do in the avoidance network, possibly slightly a product of the larger network size for the co-occurrences. When we take these hub genes and start to look at what they do, we can see here I've listed a rough functions for them all. The majority on this particular plot for the co-occurrence hub genes seem to be involved in things that we've loosely termed attack and defense. So a lot of toxin, antitoxin systems, quite relevant this afternoon, it seems, as well as a lot of them are involved in metabolic pathways, which is possibly not surprising given how diffuse metabolic pathways are, how variable they can be depending on where you live. But the main point of note, so this plot, the y-axis is out of 30 number of genes, but then when we move to this is out of 300. So actually the majority of the hub genes, particularly for the co-occurrences, are encoding hypothetical proteins. So we don't know what they're doing. So to me, it seems like we have this hypothetical network that we don't yet know what it's doing, that could be having a really large impact on the E. coli accessory genome. We don't yet know what it's doing, which to me, I find really quite intriguing. And then to go just one step deeper, we can start to look at specific gene cluster interactions. So what I'm showing you here, and I don't want to go into too much detail to avoid becoming too data dense, these are gene clusters that are involved in PTS systems, phosphotransferase systems, and they are the gene relationships within these systems. And I've coloured each of these nodes by the system that they're involved in. And so what I just want you to take away from this plot is the fact that the colours are really spread out throughout the plot. Okay, so we're not just seeing relationships purely by system. We're seeing that individual genes and individual gene clusters within a system have their own individual gene relationships, which was quite surprising to us. And so this is the only kind of real data that I wanted to present on this for you now, because there's a lot of specific gene interactions, but we should hopefully have this on bioRxiv in the very near future, I hope. And we're going to be obviously making the entire data set available with that as well. So if you have specific genes that you're interested in, please do take a look, find out what they're co-occurring with, and hopefully this can be a useful resource for people as well. And I'd just like to take a few minutes now to talk about one of the applications that we think you can do with a data set such as this, beyond simply looking at pangenomes. And so for this is a project that I'm doing at the moment, a small side project with Fiona, and then with Maria Rosa, who's at Nottingham Trent, and Lily Cummings, who works as a PhD student at the University of Birmingham. And we're interested in whether there is a genetic background that predisposes you to antimicrobial resistance, beyond simply the presence of antimicrobial resistance genes. And we're using these gene co-occurrences to attempt to answer this in some respects. So this here is a Fandango plot. Our E. coli phylogeny is on the left, and we're looking at the presence in orange or absence in purple of beta-lactamase genes in our E. coli pangenome. And then what we can do is take these genes and other antimicrobial resistance genes, put them through CoinFinder, and then we can take and look at which genes they're co-occurring with. And then we can produce networks such as this, where each of the component within the network that's coloured differently is a single antimicrobial resistance gene and the genes that they're co-occurring with. So for example, we can take in light blue at the top, we can take all the genes that are co-occurring with this antimicrobial resistance gene and look at where this cluster is found on the phylogeny. Are they particularly concentrated in multi-drug resistant strains? And then if we can do some small wet lab experiments and say, well, if we don't have these genes, does this change our resistance phenotype? And the reason I'm asking lots of questions and posing lots of questions now is because we haven't actually answered any of them yet. This is still very much work in progress for this. And so I'd now just like to briefly summarise that I've presented to you this large E. coli network of coincident gene-gene relationships in the large E. coli pangenome, and shown that they really vary with their presence or absence across the E. coli lineages, and suggested one possible thing that you can do with such a data set in terms of looking at the presence of antimicrobial resistance across the network. And so this was this work was supervised by James McInerney at the University of Nottingham, and it was BBSRC funded. Fiona, Maria and Lily I've already mentioned. Chris Connor is a PhD student at Birmingham who did some initial work with E. coli genomes, and which came out of Alan Manali's lab. So thank you very much, and yes, if anyone has any questions in the chat or anything, I'd be happy to answer them.
Thank you so much, Becky. That was brilliant. That was really interesting. As you know, I love this topic and I always have so many questions. So I see one from Mike that kind of overlaps with an obvious one that I had. Mike's question is, using the co-occurrence and avoidance network, can you identify mobile genetic elements that are common in particular genetic backgrounds? Thinking about plasmid incompatibility groups or prophage protection to superinfection. If that makes sense.
Yes, absolutely. Yeah, no, that's definitely something that we can do. It's not something that we have done for this specific paper, because for this work we've really tried to give like an overview, but it's definitely something that we've noticed that mobile genetic elements crop up a lot across the network for sure, and that's one of the things that we think could be driving, along with the shared function, is one of the things we think could be driving these relationships. Possibly the fact that you are moving around a lot, you're then almost picking up genes that you're co-occurring with because of your mobility. So for sure that's something that's definitely could be used for sure.
So that was also a question that I probably had for Chayan as well, but we can pick this up later. But especially when you both give the same example of the toxin, antitoxin being quite abundant and spread, yeah. Are those associated with mobile genetic elements? Are they associated with anything in a particular species or the way they're spread? Is there anything unique about why they're so easily spread?
Yes, so they're found relatively constant across. It doesn't seem to be linked too much with whether you're an MDR lineage or not, but yeah, we do see quite a lot of connection again with those with MGEs as well. They do seems like mobility is really interesting for that. Yeah, for sure.
Okay, well, in the interest of time, maybe we can pick this up later if anyone's got any more interest. So thank you again for that. That was really interesting, and thank you again, Chayan, as well. That really kind of merged up some really nice tools there. Okay, I'm going to hand over to Gavin to introduce our next speaker.
Thank you, Michelle. So thank you, Michelle, Laura, and Michael for arranging today's great conference. Thank you, Michelle. So thank you, Michelle, Laura, and Michael for arranging today's great conference, and to Christoph again for that fantastic tribute to Will Coogan. So for the final talk in today's session, I'll have great pleasure to introduce the first James Burgess Lecture on microbiology. And we in the Department of Biology here at York are very fortunate to have a friendship and philanthropic support of Ashley and Jill Burgess, who are joining us today. Ashley, Jill and their daughter Sarah set up the James Burgess Scholarship Programme at York 20 years ago now, in memory of their son James. And this scholarship funds a PhD project across the areas of biology, biochemistry and biomedical sciences in the department, and as most recently, this is focused around the important theme of antibiotic resistance. And this year, the family have extended their support to the department to include this annual lecture named after James. And Ashley is a Moral Fellow of the University of York in recognition of his philanthropy, a member of the University Court, and Chairman of Trustees of the charity Antibiotic Research UK. So we'd like to thank the family and the family company, the Burgess Group, one of the UK's leading animal food manufacturers, for their long support of the research in this department. So our first Burgess lecturer is Professor Ruth Massey from the University of Bristol. She's someone we've been wanting to come to speak in York for a couple of years now, so at least we've managed to get her to speak. We'll have to get her up for some Yorkshire hospitality at some point in the future. So Ruth spent her early career in Ireland, where she did her degree and PhD at Trinity College Dublin. She worked with Professor Charlie Dorman, who's a leading molecular microbiologist, on plasmid function and evolution in a gram-negative pathogen, Salmonella typhimurium, before she moved to Oxford to work with the evolutionary microbiologist, Professor Paul Rainey. Then she made a switch from gram-negative to the gram-positive world to Staphylococci, first working with Sharon Peacock in Oxford. And the Staphylococci are very familiar to us as containing the antibiotic resistant strains we hear about in the news, such as MRSA and VRSA, and are increasingly important pathogens. So Ruth gained her first faculty position at Oxford in 2004, and then she moved to the University of Bath in 2007, where she's really built a name as a world-leading staphylococcal researcher. And she then moved down the road to Bristol in 2018 to become a professor of microbial pathogenicity. And today she's going to tell us about how we can use genomes to understand microbial pathogenicity. So thank you, Ruth, for accepting our invitation and for delivering the first James Burgess lecture.
Oh, thank you for having me. Hopefully you can see my screen now. Yep. Great. Yep. And I'll just put on a laser pointer as well. Okay. Yeah. Thank you so much. What a lovely invitation. Lovely invitation. A really bad echo. Oh my goodness.
Yep.
Is that my end, do you think?
I think it's fixed now.
Okay, brilliant. Thank you. Thank you. Right. Yes, lovely warm. And I just want to start off by warning you that we live in a cottage, and I've got 3 really bored teenagers in the house and 2 dogs who'd like to completely lose their mind at anything like a pigeon lands in the garden. So I will. I'll start with that warning. Hopefully they're all on. That they've been fed chocolate and given electronics. So hopefully they'll remain quiet for a while. So yes, thank you so much for the invitation. And I've really enjoyed the talks this morning or this afternoon, and I'm looking forward to coming back next week to hear some more. What I'm going to talk to you today it's a bit of a journey, because who doesn't love a journey, and it starts with our early work on looking at could we use genomics to understand virulence and pathogenicity, through to what we're doing at the moment and what we're really hoping to do in the future with genomics. And so there's a couple of short stories within this talk that I hope you'll enjoy. Now, given the current climate, I don't really need to convince anybody of the importance of studying infectious diseases, but it's worth mentioning that, antimicrobial resistance, or AMR, was a problem long before sort of COVID reared its ugly head, and it's going to be a problem long afterwards, highlighted just this week by this Guardian article. And it's something that we really need to be mindful of. This is some of the projections, scary projections that have been made about what will happen if we don't address antimicrobial resistance. So these are some of the predicted annual death rates for AMR infections by 2050 if we don't do something about it. And it's clear that economically developing countries are probably going to be the worst hit, but as we know, we live in what we probably consider to be a fairly privileged environment of economically well-developed countries, but we're very much not safe from these AMR infections, and it's going to be a significant problem if we don't start tackling it. And the organism that I'm going to talk about today is very much a global problem, and it is Staphylococcus aureus, as Gavin mentioned. And just to highlight the problem that it represents in England alone, This graph here is the data from Public Health England, where back in 2007, it became mandatory reportable that all MRSA, so methicillin-resistant Staph aureus infections, became reported to Public Health England, so they could monitor all bloodstream infections. And as you can see, this is about the time point where we were getting a handle on it. So this is the green line here. And the incidence had already dropped quite a bit, but it plateaued in about 2011, 2012, and it really hasn't moved since. We're not able to shift this from the hospitals. What was interesting, though, is that there was a lot of observations that as the MRSA rates were going down with regards to bloodstream infections, the MSSA, so the methicillin-sensitive rates, were going up. And this is, you know, alongside all these sort of you know, infection control and hand hygiene measures that were going in the hospitals. And so this is represented, so the MSSA infections became mandatory reportable in 2012. And what you can see here is that we've had a year-on-year increase in the incidence of MSSA bacteremias in England, and this has been reported across the world now. I think in England since this time there's been a 30% increase in incidence. So we have absolutely no idea why this is. We don't know why suddenly we're much more susceptible, or there's greater populations being susceptible to MSSA infections, but it's a worry. And to talk about the type of infection, so this data is all from bacteremia, which is one of the more severe infection types, but Staph aureus is an incredibly versatile organism. It can cause really minor things like skin and soft tissue infections, so boils, carbuncles, furuncles will more than likely, if you've ever had one of those, will have been caused by Staph aureus. Styes in your eyes, if you ever had one of those, or sinusitis, that's probably going to have been a Staph aureus infection. And that's because it's present on about in the kind of nose and throat area of about 30% of the population, and it can take advantage of breaches in your immune system to cause infections. Usually these are very self-limited, you don't even need antibiotics, you'll clear it yourself. But if it gets into deeper tissue, that's when it becomes really problematic. So if it gets into the lungs, it can cause pneumonia. If it gets into the bloodstream, it causes bacteremia. It can get into your bones. It can get into, you know, cause attack heart valves, or cause endocarditis. So it really is an incredibly versatile pathogen, both with regards to the part of the body that it can invade and the severity of the infection type it causes in there. So how does it do this? Well, this is a much loved sort of cartoon image of a cross section of a Staph aureus cell, and what we have on the left hand side here is just a small number of the proteins that it expresses on its surface that allows it to bind to us. So we have elastin binding proteins, collagen binding proteins, fibronectin binding proteins. And I know these are proteins that are of great interest to many of you up in York, especially with Jen Potts up there. On the right hand side, then we have some of the proteins that it secretes, and these would be the toxins. So these are proteins that the bacteria secretes that then go out and cause damage to the host or the tissue surrounding that. So it's incredibly versatile. And what we tend to do is we clump the different strategies that the bacteria uses into its ability to adhere, conferred by its adhesins, its ability to cause tissue damage, conferred by its toxins, and its ability to evade the immune system. And this is critical as well. They're not necessarily distinct. The evasins will have some adhesins and will have some toxins within that, based on, you know, what the outcome of the interaction with the host tissue is. But there are also then some very specific proteins that are, their sole job is just to mess around the immune system to allow it to cause disease. And so in addition to all of these proteins that it has that allows it to cause disease, it has an incredibly complex regulatory system. This was from a review article that we wrote back in 2012, and this is when we were first starting to think about how we should maybe change how we look at pathogenicity. I think up to this point, we've been very focused on looking at maybe one protein in one lab strain and studying that in detail, and that's not to knock that work at all, because that's been incredibly helpful at getting us where we are. But we wanted to just see whether we could slightly shift focus and look at things from a slightly more population level. And the starting point that brought us to that was this study where we tried to map the regulatory network of Staph aureus with regards to virulence, and this is kind of what we came up with. So what you have on the outside here, A, T, and E are for the adhesins, the toxins, and the evasins. And on the inside, each of these purple blobs is one of the known regulators, or the regulators at that time. And as you come out, say, for example, AGR, a very well-known one, this is known to repress adhesins, activate toxins, and repress evasins. So you can see then what we did was we tried to capture all the information of what these regulators did to these three major groups of virulence strategies. Lots of question marks there where we didn't know what they did. And then you have in the middle then all the arrows and repressor lines just showing you what was known at the time about how they interacted with each other. So it's massively complex. And this is back in 2012. And since then, you know, I don't know that we'd even get it all onto one figure, what we know now. So there's so many loci. We've got all these genes that actually do something, that physically interact with the host. And then we have all these genes that regulate those genes. So there's a huge number of genes, and we really thought that this has got to be an opportunity where there must be huge amount of variability to be seen, and that was our starting point really. Now we started looking at toxin production because it's relatively easy to measure. So Staph aureus is well known for secreting lots of cytolytic toxins. Other toxins too, but when I refer to toxins in this talk, I'm referring to cytolytic just for simplicity. And what we have here is two Staph aureus strains, one a very toxic one that's been secreted on a horse blood agar plate, so that's the red background. And you can see here where the bacteria have grown, they've secreted their cytolytic toxins, and we've got all this lovely lysis. Compare this to a very closely related but non-toxic Staph aureus strain on the right-hand side, and we've got no cytolytic toxins. So this is the phenotype that we started focusing on. And we collected, we had lots of people who had done lots of sequencing who were very interested in giving us their strains, which was fantastic. And so we started collecting these strains and looking at how much toxins they produce. So we had these are 5 of the major clones that cause problems around the world. 22s and the 36s would be the 2 clones that cause most of the problems in UK and Irish hospitals. ST93 is a very predominant Australian clone causing problems in their hospitals. ST8s, these are interesting. These are the USA 300s, or they're often referred to as the USA 300s. And they are the community acquired MRSA. So this is this kind of relatively new, not that new anymore, but compared to the hospital acquired MRSAs, these are the ones that are causing infections in healthy people, whereas the hospital acquired tend to be limited to causing problems to sick people. So these are very interesting in their own right. And the 239s, this is a big Asian clone. And what this is here is a huge amount of work. It's about 500 clinical isolates here. Each dot represents a strain and how toxic it was. And we're measuring toxicity here as the percentage. So we culture THP1 cells, mix them with the bacterial supernatant, and look at how many cells survive that exposure. So those toxins will be in that supernatant. And so each dot represents how many, the percentage of cells that were killed by the bacterial supernatant. The first thing I wanted to draw your attention to is this dotted line here, because what we observed really, really quickly was that there was huge clonal differences. We had some clones that were so toxic, their supernatant was so toxic, we had to dilute it. So these are these 3 clones to the left-hand side. We had to dilute those supernatants down to 30% because if we use them at 100%, there was just nothing alive and it didn't allow us to measure anything. Whereas these guys here, the 239s and the 36s, we could use them at 100% supernatant. And if we diluted them down to 30%, nothing died. So this is just the first thing to point out, that there were very significant clonal differences between how toxic they are. But the next thing that you need to see is obviously the huge range. All these dots represent one strain, that even within the clones, there was huge amounts of variability in the clones. And this is something that we kind of have been pushing quite hard, because we tend to be a little bit lazy, possibly not restricted to the Staph aureus field, but we often talk about clonal phenotypes, like they're common to the whole clone. And this very clearly is showing you that we really shouldn't do that, certainly not in relation to toxicity, because yes, okay, we did have to dilute some clones more than others, but actually the variability within the clones is so huge that we really need to be careful about when we're talking about clones. So where does genomics come in? Well, as I mentioned, all of those collections of 500 isolates had all had their genome sequenced. And so we were able then to use GWAS, or genome-wide association approaches, which is a statistical test really, to see if we could identify new genes that associated with whether a strain was highly toxic or not. And this is a typical product of GWAS, where you have a Manhattan plot, and what you're looking for. So what this is for those of you not familiar, on the x-axis is basically the genome, so the origin of replication all the way through to the terminus, kind of laid out. And on the right hand, on the y-axis is the probability of polymorphisms in that locus being associated with your phenotype, in our case, toxin production. And what you're looking for is these kind of skyscrapers. I'm pretty sure that's where the name Manhattan came from. Because what that means is that there's clearly a gene, for example, this one here, where we've got this collection of polymorphisms popping up that are all associating with this, and another one here, for example. And that basically suggests that there's probably that the gene encoded at that site is probably involved in toxin production because there's so much variability in it. And we've done this, and this is just some of the genes. What we tend to do then is to prove that they're causative, because obviously there's stats, stats as a starting point, but really making the mutant is the ultimate endpoint for us anyway. And so these are just some of the genes, the brand new toxin associated genes that we've found so far. The top 4, we're kind of working our way down this list. So we published these top 4, and we're working on these ones now. So it's been a very, it's worked really well for us, and it's allowed us to identify all these brand new regulators of toxin production. And I'm going to give you a snapshot of just one of those proteins and what we're doing with it and how we're characterizing it further. So this is a protein that I'm hoping I might get some good ideas about what it's doing, because it's a very interesting protein, but it's a bit of a mystery to us. So MSPA protein, associated by GWAS through toxicity, we knocked it out to prove that it was involved in toxin production. So what it is, it's predicted to be a membrane protein, small 105 amino acid protein, 4 transmembrane domains. So we're pretty sure it's a membrane protein. When we knock it out, so JE2 is our wild type strain here. When we knock it out, we completely lose the ability to lyse the cells, and this is complementable. So this is putting it back in on a plasmid and switching the plasmid expression system on. So we could complement it and verify it. I mentioned AGR earlier. This is one of the major toxin regulatory network loci in Staph aureus, and it's always the first go-to. If you have an effect on toxin production in Staph aureus, it's always wise to go to AGR first, see if that's involved. And sure enough, when we looked at that in 2 different backgrounds, so we looked at JE2, which is one of these USA 300 MRSA strains, and we also looked at SH1000, which is an SSA strain. In both situations, when you knock the MSPA gene out, the AGR system does not get activated. So we got AGR measured on the y-axis here. So this was all involving AGR. So we're kind of going, okay, well, let's see, why does it do this? Okay, so certainly without MSPA, the proteins don't produce cytotoxic toxins, and this seems to be mediated through a lack of activation of the AGR system. We decided to do, we've got some really good facilities in Bristol, and we decided to do some tandem mass spec to see if we could work out why the AGR system wasn't working, or what else this MSPA protein might be doing. And it's produced a rather sort of intriguing list of protein production differences that we're kind of working our way through. The first was that staphyloxanthin biosynthesis was, the proteins involved in that were affected or were differentially expressed. And staphyloxanthin is the gold, is responsible for the characteristic gold pigment of Staph aureus. And certainly when we then had a look at the bacteria, the MSPA mutant, so this is just an Eppendorf of the bacteria spun down, so you can see it's an upside down Eppendorf. You can see here wild type Staph aureus nice and gold, MSPA mutant not very gold at all. So our proteomics thing certainly backed up by when we looked at the bacterial cells. Now staphyloxanthin is really important to the bacteria because it's responsible for a lot of innate immune defense mechanisms. So first of all, rather than just relying on color coding, we actually quantified staphyloxanthin, and sure enough, in both our JE2 strains, so staphyloxanthin measured on here, in our MRSA background, we had a significant difference, as well as in our MSSA background. Staphyloxanthin production is definitely off. And when we then looked at some of these innate immune responses of the bacteria, for example, this is survival in oleic acid, which is a fatty acid that has antibacterial properties. The MSPA mutant was really not very good at protecting itself from this. We also looked at defensin, so this is human defensin 1. Again, the MSPA mutant was really not very good at protecting itself compared to the wild type strain. We also looked at survival following phagocytosis, and again, the MSPA mutant was a bit rubbish at this. And we decided these are all quite different mechanisms. So the survival to oleic acid would be quite different to survival to defensins, which would be quite different to survival to phagocytosis. So we just decided to look and see how the membrane was looking really by just growing the bacteria in STS to see whether this was just a general, the membrane's just a little bit rubbish. And you can see here the MSPA mutant in the grey bars here. As we increase the amount of STS, the MSPA mutant just really couldn't survive very well, even just from exposure to really mild amounts of STS. So in the absence of MSPA, the mutant can't protect itself from innate immune attack. We had a bunch of proteins that were involved in iron uptake and iron efflux upregulated. And this was intriguing. So what happens with iron, with lots of bacteria, not just Staph aureus, is it's needed for a lot of metabolic processes, but it's quite toxic if it gets too high. So what happens is there are a bunch of proteins that bring iron in, so heme iron in from the environment. There's a regulatory system that detects the levels of iron in the cytoplasm. If it gets too high, this activates an efflux system, the HRTA system, which will then pump the iron back out again. And we wondered whether there was some problem with iron homeostasis in the MSPA mutant. And we decided to test this using an antibiotic called streptonigrin. So streptonigrin binds to and breaks down DNA, but only in the presence of iron. So what you can do is use it as a proxy for intracellular iron levels. So if your bacteria suddenly start dying more in the presence of streptonigrin, it means that it probably has more iron intracellularly. And sure enough, so this is just a lawn of Staph aureus where we've dropped some streptonigrin into the middle of the lawn. And you can see here, this is our MRSA strain. This is what the zone looks like when MSPA has been knocked out. So we get a bigger zone of clearing, basically, because the streptonigrin is killing more of them because there's more iron. And the same happens with our SSA strain, the SH1000 background, much bigger zone of clearing when the MSPA mutant was knocked out. So in the absence of MSPA, the Staph aureus can't control iron homeostasis. Well, one of the other observations from the proteomics, I'm nearly at the end of these other observations and what we did next door, as I promised, but the penicillin binding proteins were increased, which would suggest us so these are really important in cell wall biosynthesis for the Staph aureus, and they are targets for antibiotics such as oxacillin. And so we wondered if maybe there was some difference in resistance to this. So this is an oxacillin. It's a bit of a blurry picture, I apologize. So this is the wild type strain. We have a disc of oxacillin in the middle there, and we can see clearance here where the oxacillin has diffused in and killed the bacteria. But this is the MSPA mutant, and you can see there is clearance, but it's not as it's much smaller and it's a lot hazier, and that's because the bacteria were able to cope reasonably well, certainly relative to the wild type strain. So in the absence of MSPA, the Staph aureus is actually more resistant to beta-lactam antibiotics. And with all these phenotypes, we decided that it's likely to be very hard for this organism to cause an infection, but we wanted to test this. And so we used a skin infection model. So these are 6 mice that have had a hole punch in their skin, and the bacteria are added to this hole punch. And what you do then is monitor the development of the abscess over 6 days. Wild type up here, you can see over the 6 days the abscess gets bigger, and we included AGR as a mutant because it's well established as not being very virulent. And you can see here they do get an abscess, but it's nothing compared to the wild type strain. Whereas if you look at the MSPA mutant, it just never got hold. The mice never developed an infection at all, which is unsurprising really given all the in vitro phenotypes that we saw. And this is just reflected down here. We have abscess area here, the MSPA mutant being absolutely rubbish at causing an abscess. And we also measured colony forming units per milligram of tissue. And again, the MSPA mutant really was very attenuated. So this is a summary of some of the, it's very descriptive, this study, and that's kind of where we are at the moment. But this is just a summary image of all the things that are wrong in this MSPA mutant. And the one common thing really is that all of the processes that we've looked at involve the membrane, and this is a membrane protein. And it kind of suggests that, given that the membrane seems to be key to this, that perhaps the MSPA mutant, its membrane is in some sort of chaotic stasis. And we decided to actually have a look. Let's see if we could see this. So we did some electron microscopy, and this is a wild type Staph aureus cell. And you can see we've got nice smooth. So we've got the outer sort of cell wall, and then you can see the white line here is a nice smooth membrane, and then we've got the cytoplasm. So this is what a nice healthy Staph aureus cell looks like. This is what happens when MSPA is knocked out. You can see that the membrane is completely ruffled, and we even have some very crazy, strange invaginations, really sort of wavy formations going up in the membrane. And this is another one here where the membrane started looking, you know, like real invaginations. And so it's very clear that the membrane is complete chaos in this mutant, which would explain why we're getting so many membrane-associated phenotypic changes. And we're currently in the process. So we've kind of published a description of this, but we're still in the process of trying to work out the mechanisms. And I like presenting this because I always get great ideas from the audience about what they think might be going on. So anybody has any ideas, just stick them in the chat. But we're currently doing some assays. We're doing some pull down assays to try and see if there's any partner interactions and other experiments. But yeah, we're in the process of trying to work out its mechanism. So that's kind of sums up, I guess, the type of stuff that we've done with genomes in terms of identifying and then characterizing new important genes. But we really wanted to take the next step with this approach, and that is to look at whether it can help us understand patient outcome as opposed to just in vitro lab phenomenon. To do this, we got a collection of about 300 strains from the 2 clones that caused problems in hospitals in the UK that I mentioned further. And these came from Sharon Peacock, so my old postdoc supervisor. These were all from cases of bacteremia. And we had a decent amount of information on the patient, most importantly, whether they had any underlying health conditions or whether they died from their bacteremia or not. So we had 30-day mortality rates, and so we had the genome sequences. And so we did a lot of phenotyping. We looked at how toxic they were. We looked at whether they could form a biofilm or not. And we basically modeled all of that data together to see whether we could predict whether the patients survived their bacteremia or not. Because obviously if we can look at that and if we can model this accurately, then it means we're doing a pretty good job at knowing what is causing the patients to die or not. Now, the modeling approach that we use is called random forest, and it's a really useful one because you can give it as much information as you want. Some modeling methods overfit the data, and you get kind of false warm and fuzzy positive results. But random forest is better at not doing that effectively. This is one of the outputs that you get from it. And again, I'm going to assume that not everybody will kind of be familiar with these type of graphs. It's a ROC curve. And what it is on the x-axis, you have the false positive rate of prediction. So as I said, we're predicting 30-day mortality rates. And on the y-axis is the true positive rates. And so what you want is, well, basically, if the data that you give the model is rubbish, as in it's nothing to do with mortality at all, you'll end up with a dotted line like we have across the middle here. And that basically says the data you've given it is accurate as often as it is inaccurate. So it's 50-50 that your data is good or not. What you want is your ROC curve to go up to the left, up and to the left. So basically, this is what we're hoping for here. And what you can then do is give a number to your ROC curve. So if you imagine the area of this box is equal to 1, that middle dotted line would have an area under the curve of 0.5 because it's half the box. So you want area under the curves or AUCs above 0.5 and as close to 1 as you can. So what we did with this data is we gave it to we modeled it in different ways. We gave it just the phenotypic data. So what we mean by that is the bacterial toxin production, biofilm, antibiotic resistance. Or we gave the model just the patient data, so age, comorbidities, and what have you. Or we gave the model just the genome sequence data, or we gave it all of the data altogether. And what you can see here is that when in this sort of really bright blue line, when the model could pick from all 3 categories was when we got the best predictions. So there was some useful information in all 3 categories here, but actually being able to pick from all was best. And we got an area under the curve of 0.184, which we were really pleased with. This is one of the other ways you can look at the data, and this is where you need to imagine here the quadrant. So the top left-hand quadrant is where the model predicted the patient to have survived the bacteremia and where they had survived the bacteremia. So accurate prediction of being alive, that occurred in 78% of the cases, which we're really pleased about. And the bottom right-hand quadrant is where the model accurately predicted the patient had died from their bacteremia. Again, we got that right in 74% of the cases. So we were really pleased with this. But obviously, being a bit of a molecular cellular biologist, I wanted to know, well, what? Okay, that's great. That information was useful to you, but what did you pick out? What did the model say was important? Well, the first thing that was reassuring to see was that toxin production. So this is a measure of relative importance that the model has placed on the effect it had and the ability to predict. So good to see toxin production up there as the most important feature. And we'd really expect this. So you know that if you've got something, if you've got a bacteria in your bloodstream and it's producing lots of toxins, it's not going to be good for you at all. Other things that were really reassuring was CCI class. So this is the Charleston Comorbidity Index. So this is basically how sick they were and patient age. These are two things we'd expect to see, that, the older you are and the more underlying health issues you have, the more likely you are to die from your bacteremia. So it was good to see these coming up, kind of real proof of principle. But all of these genes here, so where these have allele and a dash and a number, they're basically genes that have no known function. And they were coming up as being really important to patient outcome. And so this really conferred, so this one here, Kappa, capsule production, this was good to see because we know capsule is important for lots of bacteria, but we have all of these 4 genes here that have no ascribed function, but being very important to patient outcome. So that was really interesting to us, and we're kind of trying to work our way through studying these genes. We also looked, that data now was from the CC22s, and we had to separate them out because genetically they're very distinct. We did the same thing with the CC30s. And again, when the model, again, this bright blue line, when the model could take all the data and pick from each category, it did the best job at predicting. So area under the curve at 0.81. Oh, again, good predictions, 80% accurately predicted alive, 64% accurately predicted dead with this. But when we looked at the features that were giving these accurate predictions, it was a completely different set of genes to the CC22s. And in fact, very few of them have been characterized at all. Toxin production didn't come into it. At this point, I'll just remind you that these are the clones that I mentioned at the very beginning of the talk. The CC22s was the clone that we needed to dilute down to 30% to do any assays with it. So that's a very toxic one, whereas the CC30 one was not very toxic at all. This is the one that we used neat. We didn't dilute the supernatant. So there's very clearly clonal differences in how they're killing patients. What we found as we do work on these genes is that the CC30s seem to have gone for a very defensive strategy. So they've invested heavily, rather than trying to kill the host through kind of an offensive toxin-based approach. They've gone for a very defensive route. So they survive by defending themselves very well, whereas the CC22s have gone for a much more offensive toxin production approach. So this was really interesting in terms of thinking about clones as having slightly different strategies to killing patients. But to bring you really up to date what we're doing now, what we're really interested in now is trying to fill in this big white gap here about all the stuff that we don't know. Why can't we predict this accurately? What is it that we need? What information do we need to find out so that we can say with 100% accuracy that I know why that patient died? It died because they were elderly, or they had this and they had that in combination with this genome and with this level of toxin production. That's where we want to be, because until we can do that, we're working in the dark when we try to develop new therapeutics. So what's missing? Well, we don't know. That's what we're trying to find out. Is it specific aspects of patients' immune status? Is it something to do with their microbiome, whether it's gut or skin? Is it drug regimes? We don't have that information. Are there interactions between known and unknown features that we need to try and work out? And that's the challenge we're setting ourselves. And I'm going to talk about now, to finish off, this study that we're really just setting up at the moment, because that's what we're trying to do with this study. So we're focusing in this one, not on bacteremia, but on ventilator-associated pneumonia, mostly because of all the infections that these bugs that we're interested in cause. This is the group of patients that we think will allow us to capture this information, because you have to do this prospectively. It's very hard to do it retrospectively. So you have to identify your patient group and follow them to see what happens to them, to see if you can capture all this information to work out why they died or not. So ventilator-associated pneumonia, occurs in approximately 30%. I think it's between, depends on what study you look at. If you end up in the intensive care unit and you end up ventilated, you have a high probability of getting a pneumonia. And sometimes it's considered that your normal flora just shifts and something starts to outgrow. Sometimes it's fomites in the ICU that gets into your lung because of it. Some studies say that the actual ventilation process messes up the microbiome and causes shifts. So there's lots of reasons about why somebody gets pneumonia that we've got lots of hand-wavy ideas, but not a lot of data to back up. Sort of bugs that cause pneumonia, well, there's our lab pet Staph aureus there being a major one, but there are lots of gram-negatives too that cause it. And what we're doing, and what the study that we're trying to set up at the moment, is we're capturing patients as they enter the intensive care unit. And our plan is, although we're at very early stages, is that we're going to get nose and throat swabs as soon as the patient enters the ICU, so we can see what they have in their microbiome before they actually get ventilated. Once they get ventilated, then we're going to be collecting what's called mini bowels, and effectively it's a small amount of saline is put into patients' lungs and then sucked straight out again, and that just gives you that sort of deeper respiratory tract microbiome information. And we're planning to collect bloods as well, obviously the causative agent at the end, and then capture clinical data. And what we're hoping to do is that we'll have multiple time points. So each patient, we'll have that early, basically before, during, and after the development of pneumonia, and we can then study the microbiome. We'll have all the clinical data, we'll have the changes in the immune system, and together we can try and then try and fill those gaps about why did that patient get pneumonia and why did that patient not get pneumonia, and of the patients that get pneumonia, why did that patient die and why did that patient not die. And this is the study that we're trying to set up, and that's with some really good collaborators of mine there on the right hand side. So we've got Mike, who's the microbiome expert, and Rachel, who's the immunologist. I'm kind of the microbiologist, and then Mario is the modeler who does all the clever stuff with the maths. And the reason I'm telling you about this is because we had set the study up and we were about to set it up and running. And we were kind of lucky, if you want to use a phrase, but we were basically in the position that when the COVID outbreak started, we were set and ready to capture samples. And so I'm going to finish off today with just this case study, which brings the whole world of gram-positives, gram-negatives, and viruses together. So this patient was entered the ICU in Bristol, 50-year-old, otherwise healthy, and he tested positive. He wasn't ill at all, he was completely asymptomatic, and he only tested because his wife was a healthcare worker. So it was part of a study of families or close contacts of healthcare workers. About a week later, he started to develop symptoms of COVID and got very ill and ended up being admitted went to hospital and was admitted pretty much straight away to the intensive care unit. Interestingly, by the time he got to hospital, he was testing negatives for SARS-CoV-2. So he had cleared the virus, but he was definitely had been ill with it. Okay, so he ended up staying in the intensive care unit for 43 days, which is an incredibly long time to be in an intensive care unit. And what happened was, within a week of him being in the intensive care unit and ventilated, he developed a bacterial pneumonia. And he was given a course of piperacillin and tazobactam and recovered, appeared to recover from his bacterial pneumonia, still in the intensive care unit, still very, very ill. They cultured Pseudomonas aeruginosa out of that. It was antibiotic sensitive, so they were happy that the pip-taz would work fine to clear this, and it did. Unfortunately, about a week after he finished his antibiotics, he got a second pneumonia. And this time they treated it with meropenem, which would be quite often, if you get a recurrence of an infection after you have pip-taz, there will be some resistance, high chance of resistance having emerged. So you would treat it with a different antibiotic, so meropenem. And sure enough, when they cultured out the Pseudomonas, it was Pseudomonas aeruginosa again. It was tazobactam resistant. So we kind of had all of these samples. He recovered. The meribactam worked fine, cleared the infection, and they took a third sample from his tube, his endotracheal tube. Not because they were concerned about him having a bacterial infection, but they were just monitoring because he'd been incredibly ill. And they managed to culture a third Pseudomonas from this, but that was resistant to both tazobactam and meropenem. So the bacteria were very clearly evolving in this patient as he was being exposed to different antibiotics. He recovered, he discharged from the ward, has ongoing problems though, and sort of falls into the category that we're kind of hearing a lot more about now of long COVID, long-term sort of condition that a lot of people are reporting. And this is part of these are the clinicians that were working on this project with us. And this is just some of the x-rays, which are kind of interesting. So this is what this chap's lungs like when he entered the ICU. So you can see that this is not what a healthy lung should look like, all full of cloud, full of white blood cells, causing problems. And this is where he was ventilated, and you can see how bad the lungs are. You can even see the intubation tubes and all the different wires he was wired up to. This is what it looked like after he had a second infection. So this is no bacteria. This is plus bacteria. This is after he cleared the second bacterial infection. And this was him at a follow-up clinic. And these dots here, these are considered the ground glass opacities. We can see better on the CT scans. But anyway, this is obviously, I just find this stuff fascinating because I don't do this really. We look at bacteria on Petri dishes. So when you see human samples, they get amazing. I mentioned we had all these samples. We were poised, we were ready to go. And so we decided, we managed to get the leftover endotracheal, so what they sucked out of his endotracheal tube from his first pneumonia. Unfortunately, the second one was lost somewhere in the lab, and we also got the endotracheal, what they sucked out of the third case. So he didn't have pneumonia with this one. This is the one where he was on the road to recovery, but we got what was left over out of the endotracheal suctions, and we sequenced them. We were able to do that without doing any enrichment. We didn't do any culturing, so we literally sequenced everything that was in the sample. No, we didn't get rid of human DNA. We just went straight in, and we were able to sequence the Pseudomonas strain, get whole genomes from that, which was great. We were able to identify the mutations that occurred that gave the resistance to tazobactam in the first strain, and then to meropenem from the second course of antibiotics. What was really interesting is when you looked at the composition from the sequence data, there was such a clear difference between ETT1. So this is where he had his active pneumonia, pure culture, nothing else. There's no other DNA in there, bacterial DNA in there apart from Pseudomonas. So it's a pure culture, pretty much pure culture in that he was recovering. There was a lot more diversity. The Pseudomonas that grew was still there, representing the green bar here, but there were a lot of other bugs there. So very clearly, metagenomics is going to be a good approach to differentiating, especially with complex infection types like this, differentiating between what is an infection and what is not an infection. So where you get this dominance of an organism compared to the right-hand side here, ETG3, where you actually got lots of bugs, which is more representative of a healthy, diverse microbiome. How am I doing for time? Okay, not too bad. Okay, right. So we also, as I mentioned, we were looking at the immunology side of things. We looked at his T cells. This is what was another really interesting side of this patient. This is what, CD4s and CD8s, two classes of T cells. What we're looking for here is when they're shifting in these FACS spots up to the top right-hand side, that's when they become activated in this FACS. I love looking at these. The immunologists roll their eyes at me when I show these because they like them in graph form, but I love seeing them like this. So this is a normal healthy age match control. Some activated CD4s and CD8 cells, but not very many. This is his T cells when he was first admitted to the ICU. So unsurprisingly, after a viral infection, he had lots of activated T cells. This was just after he was had his first bacterial VAP, and you can see the CD4s are going crazy. And this continued and continued, CD4s in particular. He just had this huge activation of T cells, and this continued and continued. And you can see here, I mean, this is not, having this many T cells in your bloodstream is not a good place to be at all, especially given that he cleared the virus already. The antibiotics had cleared the bacteria for him, and this is how many activated T cells he had, wandering around his bloodstream. And this is more how the immunologists would like to see the data in graph form, but you can see the levels of activations are through the roof of the different categories. What was really interesting, though, is that when we looked at what these T cells were responding to, we got some Pseudomonas antigen and we had some SARS-CoV antigen, and we threw in a third one, and we threw a third one in because we're looking for non-specific. So we always think about T cells as being very specific and they only get, activated to the antigens that they present. But there is a phenomenon known as bystander activation, and that is where the T cells that are responding to a different antigen will get activated in a bystander way, so non-specific activation. And sure enough, this is what we saw. So looking at so we had our our non-specific antigen showing just as much level of activation as the specific one. So that's possibly what was contributing to this massive amount of T cells. There's lots of the T cells that were not specific to Pseudomonas and not specific to the SARS-CoV virus were also becoming activated in this bystander mechanism. So, and these could well explain why this patient had these ongoing symptoms. Now, this is a case study. It's one patient. We can't really infer much about this, but I think what it shows, what it proved to us is that this type of approach could be very, very powerful at studying things if we can get more samples and if we can recruit lots of patients. And we can learn an awful lot more about the infectious disease process if we can do this. So this is the summary of the case study. I can appreciate time is dragging on, so I won't go through these. It's all fairly obvious. But in conclusion to the overtalk, we use population-based genomic approaches. We use it to identify novel effectors of known virulence types. We use it to look at how known virulence types contribute to disease. And then we look for, can we use it to predict patient outcome? Can we identify novel virulence traits? But the problem, the major problem here is that we're still a very long way from understanding pathogenicity, and we need new approaches to achieve this. And that's kind of what we do in my lab, is try to push what we can do with these type of approaches. So I've mentioned lots of people at various stages throughout the talk, but this is the group in Bristol. The MSPA people are Dora, Ala and Shauna, and the other guys work on all the various other projects. And these are the people who fund me, and I will finish on that note. And thank you very much for your attention.
Excellent. Obviously fascinating, Ruth. Some lovely little sub-stories culminating in that really, really topical and slightly frightening case study. So I've got lots of questions, but we've got questions in the chat, and I think we'll take some of those first. So if you put a question in the chat, I'll kind of read your name out, and then you can kind of ask your question if you can remember what it was. So some relate to the population genetic stuff, some relate to the MSPA story. So we'll start with Michelle. Had some questions about the MSPA protein first.
I think I was just dumping in the questions as I was going along. Yeah, back there was a really cool story. I just kind of answered some of my questions as you were going along. Obviously, you think it's involved membrane integrity, showed that really nice with the structures. But do you have any predicted structures? Have any homologs? Is it have import, export? Do you think it could be not taking up a nutrient, or do you think it could be not exporting something? Do you know how abundant it is in the membrane?
All totally brilliant questions that unfortunately we don't really have the answer to. What I can so we have some collaborators who are looking at the structure, but they're really at the beginning stages of that. It's something that is so hydrophobic, and we're not structural biologists anyway, so we probably wouldn't have touched it. But these guys said they'll have a go. So we've got people working with us on the structure. And we have tried to make antibodies to it and spent a load of money on it and failed. So we have a M-cherry fusion now, and we have the antibodies to the M-cherry. So we're doing some work on that now. We're trying to work out, does it work as a monomer? Is it, dimerides? We're trying to do pull downs to see if we can identify, does it interact with other proteins? It's not very abundant. So even with the fluorescence protein, we still have to use the M-cherry antibodies to see it. So it's definitely not very abundant. And so we can see it up by microscopy, but we start looking on gels, it's hard to see. So we're doing some work with some of the guys in Newcastle who are much better at visualizing bacterial cells than we are. They're going to help us with that. So we really don't know. We're at that real exploratory phase of trying to work that out.
It's quite striking then, precision effect on the membrane integrity if it's not that abundant, I would have thought.
Yeah. And it's in all the staphylococci that we've looked at. It's in Pseudomonas, it's in Streptococcus pneumoniae and then nowhere else. It's very strangely distributed. So whether it's a cofactor that's, working with something else, whether it's involved in secretion and it's a cofactor secretion for Staphylococcus. So yeah, hopefully we'll start getting some of these answers soon.
So I was going to ask a question about distribution, which you've already answered, but only found in cocci. So do you think it somehow might relate to the cell, the particular tension or curvature of the membrane? It might be preventing something to do with that, and if you remove it, when you knock it out, do you think the effect on toxin production is direct, or actually some indirect effect of changing something else and then that affecting expression of the toxins.
We wondered whether it was a secretion problem, but then when we saw AGR knockdown, I mean, as soon as as soon as AGR doesn't get activated, that means the toxins aren't even going to be transcribed. So we think that's an AGR effect, but the others, I mean, we've got some proteins that we just get upregulation of, and that's what's causing the chaos, because they're proteins that work in partnership with others. And so by going up, the whole stoichiometry is thrown off, I think. Yeah, I wish I had the answer to these questions.
No, it's a very, very interesting protein. So we move on to some questions around the population-based work, which I think is very interesting. Marianne had a comment, I suppose, which was also a question. You want to say your comment and question, Marianne?
Sure. Really nice talk, and it got me thinking. And perhaps you thought about it, but you didn't actually mention the options. So since bacterial genome sequencing can happen really fast, once you have this correlative model, have you considered using it, quote unquote, as a predictive diagnostic of severity of the disease, so that the clinician could actually have that, looking 3 weeks into the future type of thing?
That's where we hope it'll go. So, we can use it both for working out the basic science of the basic biology, but also then, adding to diagnostic powers. We're also combining it. We're going to do some work with MALDI-TOF data. So obviously all the bugs, when they get, cultured out, you get a MALDI-TOF spectra. And so we're hoping to work the sequence on that to build it. But yeah, that's the initial work is trying to just use it to look at the biology, but the long term would be that if we can put some sense to the genome sequence data and maybe even the MALDI-TOF data, that we'll be able to give the clinicians much more powerful tools.
Yeah, I'm sure you're aware that Oxford has been running this program on TB for quite a while, right? Combining AI with the genome sequences of every isolate, and that's starting to work quite nicely. So.
Yeah, no, definitely.
Just as a quick follow on from that, just it's more of a specific question, looking at the first plot you showed with the correlations between relative importance of particular features and patient outcome. Biofilm was second in that list, which is just kind of not a surprise, but I'm just wondering if what do you mean by that specifically? Does that mean an ICA-mediated biofilm or protein-mediated biofilm or any biofilm?
So how we measured biofilm was, given that we were kind of doing 300 isolates, was just the microtiter plate assay with, what's the purple dye? You know the one I mean, but yeah. So and what interestingly, it's a little bit of a red herring, I think, the biofilm result, because what with Staph aureus tends to happen is they either produce toxins or they produce biofilm. They don't tend to do both very well. So the biofilm result was actually a negative correlation. It was so if they didn't produce biofilm, they were more likely to kill the patient. But we think that was probably more for the fact that they were actually the most toxic ones. So if you don't tell the model that the things are potentially correlated, it will still pull both of them out. So we think the biofilm result is a little bit of a red herring, more to do with the fact that the high biofilms were low toxin producers.
Okay. Michael Bottery had a question.
Yeah, I was thinking, given how important toxins are in mortalities, particularly within some sequence types, whether antivirulent drugs could potentially be used to target these infections. But then I was thinking this question directly before you showed the example of the CC30 strain, where toxins aren't important. But then I was also thinking, people say that antivirulent drugs could potentially be useful, but would you think the selection pressure for other ways of causing mortality would allow, say, would force the bacteria down the different evolutionary pathways?
Yeah, I mean, the trick is, it's really interesting. So one of the really interesting is about bacteremia. So we look at it, so say 25% of people who get bacteremia will die, but that means 75% survive. And with children with bacteremia, even some, they don't even bother treating that with antibiotics, because they might have bugs in their blood, but they will just clear them. It's really, really interesting. It's really down to your underlying health is really important. What's interesting about the antivirulence drugs is, one of the earlier studies we did, which I didn't have time to talk about, is before we had the mortality data, we just looked at how toxic are the bacteria that get into your bloodstream compared to those that are in your nose or those that cause skin and soft tissue infections. And we found this really striking, really repeatable result that actually, if you look at lots of them, the strains that get into your bloodstream are usually not very toxic at all. So there's some weird trade-off that goes on when they get into the bloodstream. The problem is if you're unlucky enough to get a highly toxic one, then you are more likely to die from it. So it's kind of all the things that we thought were fairly straightforward, you know, high toxin production, bad, low toxin production, good. When you start picking it apart, it's not always as clear-cut as that. So I think especially if they're about talking about switching toxin production, I think they're going to be problematic, because if you start doing that, you might suddenly see a huge increase in bacteremias, because now whatever that weird trade-off that's going on, suddenly more to get into the bloodstream, which, okay, they might not die from it, but there's a lot of other things that can happen that are bad for you before you die with bacteria in your bloodstream. The other thing as well is those things are often quorum sensing inhibitors. So if you shut the quorum sensing system off, what you're going to end up is with more biofilm producers. So you could just be driving them towards being really great colonizers of your, you know, your bone or your implants, and so suddenly you just have it. You're just changing the problem without really fixing it. So I'm a little bit on the fence with antivirulence things.
Yeah, it makes sense. And it kind of reminds me of like long-term chronic pseudomonas infections generally become less virulent, have lower quorum sensing. They switch all these things off. So if you're switching off all of them, you could just be changing the type of infection rather than treating the cause. I agree. I guess.
Okay.
But is this issue maybe a time management? Because the speed at which you showed the evolution from resistance things was just incredible. So, you know, how can you get in there quicker? Can you disable the quorum sensing? Can you prevent those resistance ones from developing as quick so that you have time to treat with something else?
Or maybe together. Maybe that's the thing. You try, you know, weaken it with one thing and then kill it, you know, while it's weakened, you use something else. So, yeah. There'll be a Nobel Prize in it if you can work it.
So we had a question from Laura.
Yeah, it's kind of related to the idea of there being sort of lots of different virulence mechanisms that could be at play. And I was wondering whether you can see a difference between infections that kill a patient quite rapidly versus ones that take longer to do that, and whether that sort of separates any of the virulence traits.
Only not yet, I would say. I think we're still, that's where we want to go. That's where we'd love to see that. We'd love to be able to do that, but we're not. Yeah, we're a bit off, a bit of a way to go, I think, before we can do that, unfortunately.
So I just, maybe I can ask just one last question as the chair. I was really interested in your graph early on with the ST22, where most of them are really, really making loads of toxin, but you had a few at the bottom that were making hardly any, and obviously you've gone on to do the GWAS. I mean, so how much of that variation can you explain by the GWAS, or do you think there's other underlying mechanisms of that kind of clonal variation in phenotype?
A lot of those were AGR mutants, so these are Staph aureus will shut AGR off quite quickly. And then those other list of genes that we showed afterwards, and you know, we've never gone back to see how many, you know, of the genes that we've verified, like by knocking them out. If we go back and tick all those negative ones off, are they all gone? I've not done that actually. That's a very good exercise to do, just to see, is there more to learn that we're not seeing yet? Yeah. So I can't answer, but it's a good, it might be something we will do.
Cool. Right, I'll hand back to Michelle and the other organisers to close off. But thank you, Ruth. That was really great and some really interesting stories to follow up there. Thank you.
Thank you for the invitation.
Okay, thanks, Gavin. That was great. I think it was a really successful event, given that we could have discussed all these interesting microbial genomics for another 20 minutes. But in the interest of time, I shall allow you all to leave the meeting, that you can all leave at any time. Please join us next week for meeting 2, where we have a similar format, and again, a lot of microbiology crammed into 2 hours. That's highly accessible to everyone. So if you think it's going to be relevant, please pass it on. And again, I'd like to thank all of our speakers. I'd like to thank Michael and Laura for organising this as well. And again, I'd just like to finish on thanking Christoph for leading a tribute on Will, which was very dear to us, and I know it was difficult. And thanks for the Burgess family and Will's family for joining us today. So take care, everyone, and hopefully see you next week.